Our first class of the biological macromolecules is the class called the carbohydrates. And you're familiar with all the names of these macromolecules primarily from like a dietary standpoint. You know that in general, people talk about trying to limit their carbs. You know that carbohydrates include things like breads, pasta, potatoes. So I think that from what you eat, from the perspective of what you eat, you understand what carbohydrates are generally. But what we get to do is to talk about what their role is in living systems. So what I want to do first is to describe some of the functions of carbohydrates. One function is energy, two different forms of energy. Glucose that's circulating in your blood is the immediate source of energy. And then we also store energy in the form of a molecule called glycogen. And we have a particular level of glucose in our blood at all times. Every cell needs energy, and glucose is like a little energy packet that the blood can supply to cells whenever it's needed. But when our blood sugar levels drop, when our glucose levels drop, we've stored up glucose in the form of this molecule called glycogen, and we store about a day's worth of glycogen, which can be released whenever blood sugar levels drop. Then we store up energy, even greater amounts of energy as fat, but we'll get to that when we talk about lipids. Carbohydrates also can form structural molecules, so they can serve as structure to cells or tissues or whole organisms. Also, carbohydrates are involved in signaling between cells and adhesion between cells, so holding cells together in different types of tissues. So energy is the primary focus of carbohydrates, and we know that taking in carbohydrates gives us energy. But there are also other functions for these molecules that your cell requires, and that your cell will, the requirement for those carbohydrates your cell will fulfill by making special types of carbohydrates. The first class of carbohydrates will be the monosaccharides. And monosaccharides are the building blocks of the larger polymers. These are the monomers. They're building blocks for the larger polymers that we call disaccharides, oligosaccharides, and polysaccharides, and we'll talk about all those in a little bit. But some of the monosaccharides, the beads that are put together to make the string, are molecules that you're familiar with. You've heard of glucose, blood sugar. Fructose is another type of monosaccharide. And what I want you to notice is that these have the structure that we've been describing when we were talking about organic molecules. We've got a chain of carbon, lots of hydrogen, and notice that attached to some of these carbons in place of the hydrogen are functional groups. Here's a hydroxyl, multiple hydroxyl groups, and also a carbonyl. The carbonyl is at the end of the chain in glucose. Same numbers of atoms, but in a different position in fructose. And so these molecules interact with other things differently, like the lock and key example, even though they've got the same molecular formula, meaning that they have the same numbers of carbon, oxygen, and hydrogen atoms. This is a ketone. This is an aldehyde. And because they're carbohydrates, we actually change the ending of those words to aldose and ketose. Just because these are carbohydrates. If something ends in OSE, it means it's a carbohydrate. Now you find two structures for these monosaccharides. Monosaccharides can exist in the linear form, but they can also exist in a ring form. But no matter what form we see, this is glucose in its ring form, you always find multiple hydroxyl groups and one carbonyl group in these monosaccharides. I've talked a little bit about glucose and fructose. There's another monosaccharide that you might be familiar with called galactose. These are all monosaccharides, and they tend to have the general formula carbon for every one carbon, two hydrogens. For every one carbon and two hydrogens, there's one oxygen. So for example, glucose is C6H12O6, 6 carbons, 12 hydrogens, 6 oxygen. The ring form, we have the same numbers of atoms. We have a bunch of hydroxyl groups, and the only difference is the fact that this chain of carbons has made a ring structure rather than the linear structure that we saw in the slide earlier. I I also like this slide because it shows us that organic molecules can be shown in their abbreviated structure. So notice that wherever there's a point in this molecule, there's a carbon. And if there's not a carbon, there's an oxygen here. Anywhere you see a point in an organic molecule, not here actually, but wherever you see a point and there's no atom labeled, that means there's a carbon. Something like this oxygen will remain in the abbreviated structure. And sometimes the structures are even simplified further to just indicate that it's an organic molecule, but it doesn't really highlight the placement of the hydroxyls and hydrogens. I'm not going to ask you to draw glucose, fructose, or identify, distinguish between glucose, fructose, galactose, but I do want you to recognize a monosaccharide, know its general formula, and the types of things that identify it, like these functional groups. Now, the next class of carbohydrates will be the disaccharides, and those are formed by joining two monosaccharides. Remember that when we string together beads, that is a dehydration or condensation reaction. And we described there being this hydroxyl group and then a hydrogen, which is actually part of a hydroxyl here, that are going to participate in this formation of a bond, covalent bond, to link these two glucose monomers. The two glucose monomers linked like this in a disaccharide is called maltose. Notice we released water, so we had a condensation or dehydration reaction. The linkage between these two is now called a glycosidic linkage. It's a covalent linkage, but we call it a glycosidic linkage. Disaccharides, two monosaccharides linked by a covalent bond, and that covalent bond is called a glycosidic linkage. A couple other disaccharides you're familiar with: sucrose. Sucrose is a monomer of fructose plus glucose. Sucrose is table sugar. So every little molecule in table sugar is a disaccharide linked between glucose and fructose. Lactose, which is milk sugar, is a disaccharide, a linkage between galactose and glucose. Those are the two real familiar disaccharides. Now, oligosaccharides can be up to 20 monosaccharides linked by those glycosidic linkages, and there's lots of branching, some ring structures, and we don't spend a lot of time talking about oligosaccharides, but these are the carbohydrates that are involved in cell recognition and cell adhesion, and you find them on the outside of cells to help glue cells together and to help them recognize each other. Polysaccharides are hundreds or thousands of monosaccharides linked by glycosidic linkages. Three types to look at: starch, glycogen, and cellulose. Starch is how plants store up glucose for energy. Glycogen is how animals store up glucose for energy. And cellulose is how plants create is a form of structural protein in plants, and it's also made up of chains of carbohydrates, chains of glucose molecules. So I have here a picture of this person holding potatoes, and the picture of this person holding potatoes allows us to illustrate the three types of polysaccharides. Within the potatoes are starch granules, and starch granules are just long chains of glucose, straight chains of glucose that do not have any branching and have a special type of glycosidic linkage that can be hydrolyzed by plants in order to release glucose for energy. Also in these plants, there is a structural polysaccharide called cellulose. Again, long chains of glucose, but notice that the overall structure of the polysaccharide is one that is rigid, while over here in starch there's some bending and a little bit more flexibility in the structure. One of these is easily hydrolyzed because the glucose that's in the chain is going to be used for energy, and we use one molecule, one glucose monomer at a time. The plants can use these glucose molecules for energy, and so can we. When we eat starch, we take it in and we hydrolyze by hydrolysis reaction the glucose monomers, and then we can utilize that glucose for blood sugar or store it up for later. Cellulose, however, is our dietary fiber because when we take in cellulose, we can't actually break this linkage, and these molecules, these long chains of glucose, are held very tightly together by hydrogen bonds. And while hydrogen bonds are weak, a collection of hydrogen bonds all along the length of these three chains makes them be held together very strongly. And it's difficult for us to pull apart all the hydrogen bonds and impossible for us to break the linkages here. We can break the starch linkages, but not the cellulose linkages. And so this travels through our digestive system as fiber and unhydrolyzed. It's like insoluble fiber. You might read about some food you're eating having insoluble fiber. This is it. There are some forms of cellulose that are a little more soluble. We call that soluble or hydrolyzable cellulose, but this is impossible for us to digest. When we do break down glucose monomers in starch, we can use it as blood sugar or we can store it up as energy for later, and we build up long chains of glycogen. Glycogen is stored in our muscle tissue and also stored in our liver tissue. When the blood needs glucose so that blood can supply energy to tissues, our liver has enzymes that break apart these glucose monomers, and the glucose is released into the bloodstream. Muscle tissue also has glycogen stores, but that's primarily for when the muscles themselves lose energy, and that happens a lot more quickly than in any other type of tissue because we're constantly expending energy when we contract our muscles. So the difference between cellulose and starch, this is flexible, easy to break apart. It's also sometimes branched, and branching chains are also easier to deal with because they're not held together very tightly, like stacked together, because the branches get in the way. Another difference, this is used for structure, this is used for energy storage. The way that starch and cellulose are similar, they're both found in plants. This is the only molecule we're describing that's found in animals. It's similar to starch in that it's a form of energy storage, but the big difference is that glycogen is found in animals and starch is found in plants. So, just to summarize the types of carbohydrates we've talked about. Carbohydrates include the monosaccharides, the disaccharides, oligosaccharides, and polysaccharides. The polysaccharides are that true analogous structure to a chain of beads. And the beads would be the monosaccharides that make up the polysaccharide. We looked at three important polysaccharides: starch, glycogen, and cellulose. And all three of those are made up of glucose monomers. These are just little glucose monomers held together by glycosidic linkage. The oligosaccharides and disaccharides can vary in the type of monosaccharide that makes them up. And we also haven't covered all the different types of polysaccharides that we find in living things. These are just the three big ones. Next, what we're going to do is we're going to look at a second class of biological molecules called the proteins. And again, we'll see an analogous beads on a string structure when we talk about proteins.